Hi, this is Chase with Houston Frogs. Today I'm posting a response video to Joe's. Joe runs the Simple Bioactive group on Facebook, and he has some methodology that's very different from the norm in building bioactive tanks. So I'm addressing point by point today in this debate. From henceforth, I'm going to be actually addressing Joe directly. So Joe, thank you very much for posting your video and for the civil manner in which you're speaking in your video. This is a very hot topic for a lot of us, and I know that sometimes tempers can get pretty heated. The thing about it is we are all talking here about the health and safety of animals that we care very much about. So we're very passionate about that because we don't want these animals to suffer. We want the best lives for those animals. And of course, if somebody is doing something that endangers the health and safety of those animals, that's something that's very upsetting for people, understandably so, because we care about them. But anyways, what I'm going to do today is I'm going to go ahead and have your video playing, and I'm going to be addressing point by point. The first point that I'm going to come to is when you're talking about the amount of experience that you have in herpetology. And that's not something that I'm calling into question. Now, I do have to say that when it comes to dart frogs, those are very different experience. If you have a lot of experience with snakes or with chameleons, keeping dart frogs is a whole world different from keeping those animals. So for everybody in the dart frog hobby, we don't really necessarily consider that experience with dart frogs. A lot of herps, when you're talking about snakes, when you're talking about lizards, they don't have that osmotic skin that amphibians do, which can absorb chemicals directly into their nervous system. Amphibians, if they start getting sick, they can die within 24 hours, whereas things like snakes, iguanas, whatever you want to say, usually it takes a long time. It's a long decline, and then eventually it'll get really bad and they'll die. But amphibians are a lot more sensitive. And that's why it's so important that when you're looking at this from an experience level, you're looking specifically at the experience with amphibians, specifically dart frogs. That's what we're talking about. But with that being considered, I want you to also consider too that I'm not trying to come at this so far as using just the authority that I have, so to speak, to make my argument. Yes, I do have a lot of experience when it comes to this, and maybe I should give you a little bit of background besides what I've told you so far through text. So, I run Houston Frogs. I also run a mycological laboratory as well. I actually have one of my gar plates here, one of my Herculeum americanum. You can see this is some of the things I do. Growing out different cultures of fungi on plates, actually inoculating bags for not just gourmet mushrooms, but also for vivarium use. I also use a lot of different types of mycorrhizal fungi too, which, as you hopefully know, they have a symbiotic relationship with over 95% of the world's plants. And I also do a lot of tissue culturing of plants in my laboratory as well, which this is a Moslem tenarum. It's oftentimes referred to as a liverwort, actually. But anyways, I have a very strong background in science, and so when I look at a vivarium, I don't look at being simplistic. I look at being very complex. I got a master's in geoscience from A&M, a bachelor's in environmental science. I've worked in 4 different laboratories now. I was an environmental scientist before becoming the owner of Houston Frogs and making this my full-time job. So I want you to understand that when I'm talking to you about all these different things, I'm telling you these things because of those experiences that I've gathered. All these things that come together in biology and chemistry when it comes to a vivarium. It's, again, a lot more complex than a lot of people realize. So, but anyways, with that point being made, let me go ahead and start your video at that moment, and I'm going to start addressing your other points. Now again, you're talking about keeping them in the most humane way. This is, again, the main issue for us, is we want to make sure that we're keeping these animals humanely.
back and say, you know, the reason I'm right is because I have all of this experience. You know, that's called an argument from authority, right? That's a logical fallacy. And it isn't fair to your own.
Well, and you're right. It is an argument from authority. And this argument is being made from an authority. You've also had arguments from other authorities, pillars in the amphibian community. And the fact of the matter is that if I am asking questions about, for instance, brain surgery or about the brain, I'm going to go to a brain surgeon, somebody who's experienced, somebody who has actually done brain surgeries for many years and knows the latest and greatest methodology. If I want to know about, for instance, dart frogs, I'm going to go to the foremost experts in that field. People are going to be in it for, not just the largest length of time, the longest time, but it's also going to be the people that I see on the constant cutting edge. They're coming out with new innovations. They're people that are constantly coming out with new care guides, telling people about what kind of issues they could come across. It's the same thing if I'm looking at, for instance, any topic, engineering. My stepdad is actually an engineer. And the funny thing about it is, if I have a question about roadways or something like that, I go to him because he's an authority figure. He is someone who is extremely knowledgeable, extremely experienced in those things. I think that's really a big part of this argument here, or these multifaceted arguments that we're having, is that a lot of these experiments that are being done, that you're doing, a lot of these methods that you're trying to teach others that go against the norm, that go against already established practices, you're having all these people that have decades of experience, very well-educated backgrounds, that have kept thousands of animals of this particular subset. They're telling you that those methods are not the best. They're telling you that those methods could cause harm to the animals. And I think that should be listened to. I understand that you've done some home experimentation, and that's great. I'm all for people doing experimentation. Everybody should try to learn everything about the world that they can and be able to observe it through their own eyes. But at the same time, there's a lot of things that have already been found out. There are billions of experiments that have already been conducted. If we just learn from those experiments, then we already have a vast body of knowledge that we can build upon. But when you completely throw all that knowledge out the window and say, I'm going to have my own very limited observations based upon very limited experience in this particular field, and then you try to teach those things to others, that's the whole issue that we're having here. I agree. People can be doing things wrong for a long time. That's also why we look at people's credentials.
The other thing I thought was worth mentioning is that I get this a lot where people say, well, you're not following the scientific method. You're not doing lab experiments. You're not logging data. You don't have control. And that too is not a very good argument, because as a scientist, right, you know that there's many avenues of exploration in our world.
Now, I have to mention that, yes, you're right. That is something that very much irked me, being a scientist and being someone who writes down every variable, every result, everything that I have done that then leads to the outcome. That's extremely important, because one tiny little variable, for instance, when you're talking about the little experiment that you did with having the springtails in two different boxes and then having fungi that grew more in one than another, and you're saying the one that had more fungi was the one that the springtails were in. Well, we have to look at the species of springtails. We have to look at the particular strain of fungi that you have. We have to look at the exact humidity, the soil moisture. We have to look at how much ventilation. We have to look at the soil composition. There's so many different variables, and when you throw all those variables out the window, and then you just have your own small observations where you're not controlling all those variables, there could be 1,000 reasons why you're observing one thing in one box and another thing in this other one. And as we know, correlation is not the same as causation. You can have an experiment that you do 1,000 times and you get 1,000 different results if you don't control the variables. And as a scientist, again, that's something that I see that I want you to understand. Experiments have already been conducted on all these things that we're discussing many times over, thousands of times over. And so when you're conducting an experiment like you are, and you get your results, then you immediately teach based upon those results, it's like seeing the very, 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 very tip of the iceberg and not seeing the whole bottom half. You're seeing maybe a fraction of a percentile of what's going on. You're not seeing the rest, and the rest is what's really important here, because it can make such a big difference.
Can be explored by certainly via the scientific method, but also through, longitudinal observations, for instance. Those are valid forms of investigation that produce, quantitative and qualitative data. It's instructive, it's valuable data, and everything doesn't, if we only relied on the lab data, we would know nothing about dinosaurs. We would know nothing about the stars.
And you do have a point there that is true, that amateur scientists, amateur archaeologists have found artifacts of great value. Amateur paleontologists have found great discoveries, dinosaurs that paleontologists did not discover before. But at the same time, when they discover those things, if an amateur archaeologist discovers an artifact, they bring it to an archaeologist. If an amateur paleontologist finds a new dinosaur, they bring it to a paleontologist. The thing is that observations are great in the general public, and everyone should be trying to observe as much as they can and to learn from it. And they should bring that to those who are experts in the field. But again, we're talking about taking that very limited data that you're collecting and then teaching that to people as a factual basis for things, instead of taking that data to experts and then allow the experts to interpret it and to help you to learn what that data means.
There's many avenues of exploration. We don't want to fall into that trap. We say, I don't believe anything you say because you don't have data to support it. The observations that we do every day when we're surrounded in this environment are valid, and they're worth noting.
You're right, they are worth noting, but unfortunately without data, it is not something that is replicable. It's not something that we can really use. We need to be able to record all the information we can.
And you, I think, did a good job of breaking it down into maybe 4 different sort of areas of investigation or whatever. And one of them, you posted some videos, YouTube videos of Isopods and Springtails in nature. And I think that using nature to inform us, as sort of background information is really a good idea. But I don't think it's a good idea to allow that information to literally instruct us, because they're 2 different things. And what works wonderfully in nature in all its complexity may be not work, or may just simply be unnecessary in our captive environment. So I think we can say good, interesting, thank you, but we don't need to use that to validate our use of the other one of the other ones was about supplemental feeding.
That's something that's very interesting, a point that you make, that in nature we observe a lot of different things, but in a captive environment things are very different. And I would actually make an argument to say that in nature we observe all these complexities. We observe the interactions, the symbiotic relationships between fungi and plants, between hexapods, springtails, isopods. A lot of people know them as roly-polies, but we know these interactions that occur in nature. And once you take those interactions and you shrink them down to a glass box, the interactions in nature, I would say, are even more important to pay attention to. As we've gone along in keeping dart frogs, we found that a more naturalistic approach is the best approach to keep them. Originally, when people started out keeping dart frogs, they kept them on paper towels. And they found out that they had to change out the paper towels very frequently, or else the frogs get bacterial infections, because those paper towels didn't have anything to keep the bacteria in control. Eventually, people started keeping them on things like cocoa coir, simple substrates, so to speak. And then people found out that keeping them on substrate that has too high of a pH or doesn't drain well enough, you then get a lot of fungal issues. You then get a lot of bacterial issues. It's like where you have to change out that substrate constantly, about every 3 to 6 months. So then now we have this bioactive tank setup, or a lot of us in the dart frog hobby like to call it a naturalistic setup, because we're trying to emulate nature as much as we can. Because we realize now that those interactions between the soil and the fungi and the bacteria in the soil, the hexapods, the isopods, the frogs, the plants, all these different things really matter. And the more naturalistic we can make these vivariums, the more stable they are, and the more healthy our animals are going to be.
My take on this is really simple. As a keeper, I have a responsibility to make sure that my animals have adequate nutrition and that they have the right nutrition. If you put insects in with an insectivore, they will occasionally eat them. There's no doubt about that. But the question is, what does that do for me or for them? You know, if I'm careful with my nutrition and the amount of food, I usually put in enough food to last, all day, and I'll put in little feeder capsules under the cover, to make sure that they're gut loaded as much as possible. If I were to add sow bugs, wood lice to my vivarium, it wouldn't relieve me of any responsibility. And if I'm doing things right, it wouldn't help the frogs either, or the lizards, or anything, because it would be my job to make sure that they were adequately fed.
Now you make this point that, again, adding in those isopods, that you control the nutrition of the frogs, and that the isopods are not making any difference in that as long as you keep up with your job. Well, the thing about it is, in nature, we have to understand that these frogs acquire their vitamins and minerals through a multitude of sources. Now, that's going to be the ground that they're walking on, that's going to be the water sources that they're then going into or trampling through that are rich in minerals. That's also going to be their diet, and that's going to be where the insects that they're eating are also gut loaded with a variety of plants from their surroundings. So in the vivarium, what we really have to look at is, are we allowing these animals to have multiple sources of supplementation to be able to get the vitamins and minerals they need to be healthy? And a lot of that is looking at the soils. Like, for instance, I came up with a clay bath that is rich in various minerals to help the frogs to be able to sequester those minerals through their skin, dermally. We're also looking at diet. They're going to be foraging for different insects in the wild. They're going to be foraging for different insects in the vivarium. The thing about fruit flies is fruit flies are extremely hard to gut load. They have a very short digestive tract. They can't really hold that much plant matter. Isopods, on the other hand, actually are able to consume a lot of plant matter, detritus, things like that. They're very rich in vitamins and minerals. Their exoskeleton itself is very rich in calcium carbonate, which the frogs need not just for their bones, for bone growth, but also for neurological health. So putting those isopods in there, it's like you have a free food source and you have a free source of excess supplementation, not necessarily excess, but additional auxiliary supplementation. And when it comes to the animal's health, we need every single buffer we can put in there to our own error. And when you are not very familiar on the nutritional needs of the frogs, which I'd argue, unless you're a scientist like myself, it is very difficult to go through all the research and learn the exact nutritional needs of these animals. That's why you rely on vitamin manufacturers to make the vitamin, mineral supplements that you use. When you put things like isopods in there, that is something that is making sure that if you make a small mistake, for instance, not giving them enough calcium or even not giving them enough food, that's something that helps to buffer against issues that you could have.
And the bugs have become a needless expenditure of money, energy, time.
You say the bugs are needless expenditure. I say, so isopods are actually crustaceans, but that's okay. A lot of people refer to fruit flies, isopods, springtails, all the same, just bugs. I have people say it all the time. That's perfectly okay. The thing about it is, isopods, you can buy a dwarf white culture, for instance, for $8. You could then culture those out of almost nothing. Food scraps, for instance. You use food scraps, or even get a culture out of your vivarium. Because once you put them in your vivarium, they're going to multiply in there, and then you can actually take some of those and put them in another vivarium, and then another. So you're talking about $8, which is a little bit more than somebody spends on coffee these days, less than somebody spends on a fast food lunch. Springtails, again, most people sell those for around $8. Again, something you can culture out in your vivarium, or you can culture out on charcoal. It is so cheap to culture out springtails and isopods, and you can multiply them as many times as you could possibly or want to put into 100 vivariums, just spending that original $8 on each culture, literally as much as somebody spends on a fast food lunch. How is that any excuse to not be able to culture those springtails and isopods if it's something that is going to be beneficial to your animal, and something that is going to buffer against underfeeding, buffer against under supplementation, something that's going to buffer with springtails against different fungi overtaking your vivarium that could potentially be harmful, or with isopods also eat some fungi, or with isopods, them helping to eat waste in the vivarium, breaking it down before bacteria could proliferate, which could then be harmful to the frogs. $16 over the entire lifetime of your hobby. How is that something that you view as being expensive or cost prohibitive, or something that you would even say that you would want to save that very tiny amount considering all the other expenditures that you have in keeping these animals? Why would you say that when it's something that really could affect the health of the animals, if that's really what we care about, is the health of the animals?
Better be spent on things that do matter, because in this case, the bugs know that they make a difference in the health of the animal.
And they do make a difference in health of the animal, and again, we've proven that many times over.
And that was sort of divided into springtails and isopods. The springtails, I did an experiment where I divided, I had 2 tubs, one with a bunch of springtails and a bunch of sawbugs, and one that just had potty mix right out of the bag. And then I put my dog's poop, my poor dog, I cut it up and put it in the tub and covered him, and I watched what happened over the course of some weeks. The feces in both the tubs decomposed at the same rate. They both decomposed, which told me that it was the bacteria that was doing the decomposition and not the bugs, not the isopods. I did a similar kind of experiment in this vivarium back here when it was brand new, and I buried, believe it or not, a frozen dog chick, a bat that I found, a piece of dead fish, like heart fish and heart and snake poop. I buried it all, and within 2 or 3 weeks, it was all gone. And with nothing but the bacteria.
Well, let me address these experiments that you conducted with your with the dog poop and with the bat and the other things that you buried. The thing about it is, again, this is where it goes back to the way that you design an experiment. You have to look at all these different variables. For instance, a variable that you didn't take into account was the mass of those types of waste is exponentially more than the output from a frog in a vivarium. The other big thing about it is you're not taking into account that the quicker that you're able to get rid of that waste, the less chance you have of your amphibians getting some kind of a bacterial infection, which they're very prone to because their skin's osmotic, and they're able to actually absorb that bacteria through their skin. So when your frogs are actually trampling over that waste, that then is full of bacteria which are decomposing that waste, then you're risking your frog getting bacterial infections. Whereas when you have isopods and springtails that more quickly get rid of that waste than what it takes the bacteria to get rid of it, then you're reducing the likelihood of disease. Another big thing is in your experiment, you buried those items. Burying them a lot of times makes them inaccessible to particular species of springtails and isopods, depending on how deep you bury them. So when you do that, you are relying primarily on the bacteria and earthworms and things like that, depending on what you have in the soil surrounding them to break them down. In the vivarium, we are talking about waste on the surface of the soil. We are not talking about burying it. We are not talking about composting it. We are not talking about undergoing any kind of heat which accelerates decomposition. We are talking about a completely different thing. So those experiments have no valid data when it comes to dart frog vivariums. I hate to say that, but it's true. That is not valid data that we can use for dart frog vivariums, because the experiments and the variables in those experiments do not align with what's actually occurring in a vivarium.
So when we look at isopods, we know that they're deadly leaves and breaking leaves down to an extent. We also know that the bacteria will do that, if we wanted to. Well, the bacteria will do it regardless of whether we wanted to, but depending on the conditions we create. If you'll notice in that really dramatic video that you put up, the side-by-side comparison, all of the decomposition was happening right at the surface level, right? None of it up here was happening. And that's fine. The bacteria that exists in our terrariums will do that. It will decompose those leaves. But the question is, why do we want that? That's the overarching question. What good does that do?
Well, you ask what good decomposing the leaves does in a vivarium. Well, the thing about it is, in the rainforest, an actual rainforest, which I've been down to, I've actually been down to Chiapas before, because I used to run an amber mine down there. It's one of those side businesses that I had. Most of the nutrients are actually locked up in the leaf litter. And when that leaf litter is decomposed, those nutrients are then returned to the soil. This is why oftentimes in horticulture, people will take leaves that fall from the trees and they'll actually mulch them under before planting, because those leaves break down. They're a very rich source of a lot of nutrients, a lot of nitrogen, things that plants need to grow. So when those isopods, for instance, are breaking down those leaves in a vivarium, that's actually adding nutrients that then helps to fuel the plants on top of the poop from the frogs and the fecal matter from the isopods as well. And then when we put more leaves in there, that's like putting in more natural fertilizer, I guess you could say, that helps to accelerate plant growth.
I hear people say, well, they clean up the terrarium or the vivarium. It used to be terrariums. And that's true to a small
And you say it used to be terrariums. Actually, a terrarium is just the plants. A vivarium, viva, Latin for life, means you have a living organism in there besides just plants, something that moves around. And that could even be isopods or springtails quite also. So you could call it a vivarium when you add those in. Or animals. Animals are particularly what we're referring to. But it's semantics.
Except they do one thing. We have to ask ourselves, why is that important? Why do we have to go out and buy bugs and try to maintain them in our vivariums? What good is it that they bring?
Well, and we don't have to maintain the bugs in our vivariums. You literally put in the springtails, put in the isopods, and then they literally eat upon the decaying matter. So there's no maintenance whatsoever to take care of isopods or springtails if you don't wish to.
We know that bacteria can do the same thing. I guess the idea is that it cycles nutrients, but again, the bacteria is much better at cycling the nutrients than the bugs. I mean, maybe.
I mean, you say that bacteria is much better at cycling nutrients than, quote, bugs, but the thing about it is something where everything works together to decompose organic matter. It's isopods, it's springtails, it's millipedes, it's bacteria, it's fungi. All these things work together because each one decomposes matter in a different way. And they all work together to be able to return those nutrients that are locked up in that matter to the soil that is then available for the plants to be able to consume. So you can't just take one small aspect, pluck it out, and say, we're now going to make this in charge of everything. You really need multiple facets to make sure that that matter is broken down in the most efficient way. And also, it's something where we don't necessarily want particular strains of bacteria to proliferate to such a high extent, because it can really make the frog sick. It's a vector for disease, having wasting in there for so long and relying primarily on the bacteria to break it down. Well, and you say all they do is they make big poop into small poop, but that's not true. Any horticulturist knows, for instance, that earthworm castings, where earthworms are actually going through the soil and they're breaking down organic matter in the soil and then leaving behind their castings, their feces, that matter that they're leaving behind is actually a lot easier for plants to absorb, a lot easier to be then broken down into those nutrients, easy for plants to consume, than it was originally. And that's why, again, horticulturists use worm castings, or you could even use isopod castings, so to speak, which we do in vivariums, using isopods in our vivariums. It's another benefit of having them in there. There's just really not anybody that cultures isopods on such a huge scale that they could use their castings as fertilizer.
So we're dependent on the bacteria anyway. If we want that nutrient cycle, I agree it can be useful in many cases. And the final thing I think you talked about was the aeration of the soil. Let the bugs get in there and provide oxygen to the roots of the plants and oxygen to the bacteria that's working. And, you know, my feeling about that is that, first of all, none of the bugs I have they all stay in the surface litter. There are species who may grow. But if you're using the right substrate, which in my opinion is a potting mix. Now, if you go to University of Massachusetts, they have this Center for Agriculture or something, and they say there that the potting mixes are designed by professionals to maintain air pockets and or to ensure that plants have access to oxygen. That's what it's designed to do. And the horticulture industry is a multi-billion-dollar industry that depends on that trade of that substrate. You can compact anything, any substrate you can compact, and you hold the water. You can.
I'm going to back that up. Let me go ahead and address this. So you're talking to me first about the isopods and the springtails, and you don't observe the burrowing yourself, but we do know factually that isopods and springtails do burrow. They do cause bioturbation. They do help to aerate the soils. I don't really think that's something that can be debated, because it's just a fact. We just know it. It's something where some species of isopods burrow more than others. Dwarf white isopods, or dwarf purple isopods, of course, are going to be burrowing a lot more than Porcellio scaber, for instance. There's some species that like to be on the surface a lot more. Springtails actually dwell both on the surface and in the soil itself. Both of them are extremely important for aeration. But then you're talking about how this potting soil is designed by these multi-billion-dollar companies. Well, it is designed as potting soil. It's designed to be put in a pot. It's designed as something that, yes, it has the perlite in it to keep it light and fluffy. It's also designed to hold on to water as well, but it's also designed with chemical fertilizers that are actually derived from petroleum byproduct. And that's something that could be extremely harmful, and is extremely harmful to animals, especially those that are able to absorb that directly through their skin, like dart frogs. And those potting soils are made in mind that they're going to have insects, they're going to have crustaceans, they're going to have hexapods dwelling within that. Because honestly, I don't think that you can put a potted plant outside or even indoors and then not have those start to inhabit that pot and start to start to go through that soil, aerating it. Those springtails, of course, I've had potted plants inside that they just turn up all of a sudden. It's one of those things where you could have one even on your sleeve. I've actually cultured many different types of springtails before, and I'll literally do everything I can to change clothes, to wash off everything that I can, and still have one particular species of springtail, the temperance particularly, that will get into every other culture of springtails, which is really, really annoying sometimes. So I can tell you that those potting soils are made with having in mind that they're going to be inhabited by these organisms. They're also made having in mind that you're going to have a drainage hole in those pots. And as any good horticulturalist knows, if you don't have a pot with a drainage hole in it where the water can drain through the soil, then the roots are going to suffocate. The water is going to stagnate in there. It's going to drown the roots of the plant. The plant's going to die. So that's really important to keep in mind when you're talking about how these potting soils are designed. They're designed for, like your experiment, growing tomato plants. They're not designed for keeping animals on. That's what other substrates, like from us at Houston Frogs, or from the Bio Dude, or from others, ABG, originally designed by Land Botanical Gardens, it was designed with having in mind that animals were going to be on it.
That's what it's designed to do. The horticulture industry is an industry that depends on that trade of that substrate.
Yes, and their trade is providing substrate that is going to grow vegetables, not to be keeping animals on, and not to be growing South American tropical exotic plants on. Vegetables, particularly ornamentals, that's what's designed for.
Any substrate
And you say substrate compacts through overwatering, and yes, water is a form of transport that helps the particles to actually compact. And it's also going to be anything that's walking on top of the substrate, but soil naturally compacts over time just because of gravity. It's just something that occurs. And that's why oftentimes people will have to go and till up fields. They'll have to go and have their lawns aerated, where they actually have machines that go and poke little holes in them. Keeping the soil aerated is so important towards having very healthy plants and having a very healthy soil biome. And funny enough, you say full of fungus. When it compacts, fungus actually respirate. They actually breathe oxygen. They actually give off CO2. So when you compact the soil so much, you won't even have living fungus in there. There won't be springtails. There won't be isopods. There won't be fungus. There's just going to be bacteria, anaerobic bacteria specifically, of which most are going to be toxic to dart frogs. And you say stop watering so much, but the thing is, most people who keep dart frog vivariums do not overwater. They're very careful about how much water is going in the vivarium. So I don't think it's just a overwatering issue. I think it's an issue of you really do need to have that substrate system, and you really do need to have something that is aerating that soil physically for you. And without you going in and overturning the soil yourself frequently, then having isopods and springtails are a great way to help aerate that substrate.
And I'll cut you some video here and show you that even after many months, if you don't abuse the substrate by putting too much water on it, you don't saturate it, you don't drive all of the air pockets out of it. But even if you do, if you have something like a potting mix, my potting mix doesn't drain, right? But it evaporates. And you can actually watch it happen on one side of the glass enclosure. You can watch the substrate slowly evaporate, providing humidity, which is desperately need, trapping humidity in surface, covering leaves and stuff, creating all the multitude of different microhabitats within the vivarium.
And you're right, water does evaporate, and that does help to create humidity. But another important aspect too is that when you have water percolating through the soil, it's also going to help to carry waste with it as well. That's one part of waste removal, ensuring that the nitrates, nitrites, ammonia, urea, things like that in your soil are not at too high of levels. A lot of that is going to actually wash down to your drainage layer, what's not been already taken care of by fungi, bacteria, isopods, springtails. And then if you have water in that drainage layer, there's actually beneficial aerobic bacteria that will grow just like you have in a fish tank. They'll then help to cycle those nutrients. They'll help to use those nutrients to where they're no longer harmful.
So that, the idea that the bugs provide the aeration and the substrate is it's not necessary, and I will cut to another video that shows you why, examples of why that isn't necessary. So I guess I went over that really quick, but the important thing is that every aspect of bugs in the vivarium that you bring up are sort of what I would call, they're certainly optional. They're not necessary by any means to have a healthy vivarium. If you enjoy them, by all means, incorporate them, enjoy the whole aspect of the hobby. But in no case are they providing benefit to our animals. And in no case are they providing benefit to us.
Well, you say that they're optional, and you say that they don't provide a benefit to the animals or to us, but I could say that a seatbelt's optional, or an airbag's optional in my car. I could take those out. I don't need them each day. God forbid there's a car wreck. I could say that having a fire extinguisher in my facility is not something that's necessary. It's optional, right? But if I ever have a fire, then I'm going to need a fire extinguisher. The fact of the matter is, we have them in there because we are not perfect, and there are a lot of variables at play, and they are a buffer. They are a buffer to fungi potentially taking over that could be harmful. They're a buffer to the proliferation of bacteria because of the buildup of waste. They're a buffer to underfeeding. They're a buffer to undersupplementation. They are a buffer to your substrate becoming too compacted. And again, we said that they are as cheap as a fast food lunch, something that's going to last you the lifetime of the hobby, many, many years, depending on how long you want to be in it. So why would you not go with an option like this? If somebody was to tell you, oh, it's only $5 more to have seatbelts installed in your car, would you say, oh no, that's okay, I don't really need them, I don't think I'm ever going to get in a car wreck? That's just not how these things work.
So rather than go down that path of that sort of needless complexity, that sink of time and money and energy, leave them out. Use a substrate that's designed well. Get rid of that misting system. And we'll talk more about misting and water and the vivarium. And I know you want to talk about substrate. We'll cover those things in another segment of this. But that's what I would have to say about the bugs for now. Okay. You can either rebut this, you can come back and say that's it. Let's move to the next topic. Whichever you want to do. You get some finer netting underneath it to keep the fruit flies in. And you can see what the humidity is. So what happens when you don't have a drainage layer? What happens to the water that gets added, right? It gets evaporated. And it gets trapped under objects in the vivarium, so that under this plant, it's more humid than it is on this rock. And under this slab bark, it's more humid yet. So the humidity doesn't need to come from constant misting that makes the air in the middle of the tank humid, where no animals exist. It just needs to come up naturally, evaporate from the substrate. We never put enough water in here to let
Well, see, that's another point too. I see that you have a full screen top on there, and you're saying that you don't need that high of humidity in the middle of the tank, quote, where there's not animals. But the fact of the matter is, most people grow plants in their vivarium that do need high humidity or they will desiccate. And those plants also help to add a buffer against undermisting, I would say, because they're going to be transpiring during the day, giving off not only oxygen but also water into the air. They're going to help to raise the humidity. But your frogs too, if the humidity is too low, it will stress them out, and then they will hide in those spots where the humidity is higher, whether it be burrowing under a leaf in the substrate, whether it be going under that piece of cork bark. They're going to be going where they're not going to be desiccating, because since they have this very osmotic skin, it's sort of a double-edged sword. They can absorb water through their skin, but they can also very easily and very quickly lose water through their skin, which is why if your frog escapes in your house, which is typically 30 to 40% humidity, then they're going to desiccate in a matter of hours. Then you're just going to find a little raisin on the floor. So it's incredibly important to have high humidity throughout your vivarium, not just in those hiding places, but throughout the entirety of it. Not necessarily 100%, but something that's in the range of 70, 80, even 90%, because that's something that's going to allow for evaporation, yes, but it's also going to make sure that your frogs are not stressed out because they're losing moisture through their skin.
At the bottom, and we never let it get wet. So there's no fungus, there's no gnats, there's no odor.
You say there's no fungus, but I guarantee you, being someone who runs a mycological laboratory, there is fungus in your tank. There's fungus everywhere, because even when you breathe, you're breathing in and out fungal spores. And there are fungal spores flowing around everywhere around you. I guarantee you 1,000% there's fungi in there. You're just not seeing the fruiting bodies.
Just balance the water and the substrate. Well, this is the important relevant part. The reason you don't need bugs here is because as the water evaporates out, the air pockets return to the substrate.
You know, as the water evaporates out, those particles, because of gravity, are going to be constantly moving downwards. You may not notice it for a while, but it will compact over time. By the way, I actually did go for a second master's in geotechnical engineering, which I unfortunately did not finish, but again, from that standpoint, I can tell you it does compact over time.
Breakaway number 2. I wanted to just show you that we talk about these are the leaves that have been here for months now. We can see that they are decomposing to a small extent, but there's no reason to have bugs do that. There's nothing wrong with that substrate at all. It's nice and light and airy. It's going to stay that way. All the way to the bottom where the moisture is. But no bugs needed for aeration. Right? He's happy about this. Breakaway number 3. This is a vivarium that's a few months old. And hopefully you can see it. There's air pockets here all the way down to the bottom. I'm not sure that's going to show on the video, but I guarantee you, if there weren't air pockets, these plants would die in here. That's just the way it is. And the fact that the plants don't die is a clear indicator to you. As the keeper, that your substrates are in good shape. You can see that the pockets here, air, all through here, it's not a bug. No bugs needed. So let's put this out of here. You can see how Springtails and Isopods operate. You've got lots of Springtails in here. You can see lots of Isopods. Very, very healthy environment for these bugs. Again, this is miracle grow potting mix. Just some sphagnum put on top, and then a layer of leaf put on top of that. What I want to show you, look at this. This thing is loaded with Springtails. And then look at all this fungus. Right? Fungus grows with the ventilation space, and when the moisture content level is too high. So that's what fungus problems are, and Springtails are not needed in any way to address that problem.
Okay, so that was actually cobweb mold that you had in there. And yes, Springtails take a while to break that down. But they will eventually eat it when they're in large enough quantities, large enough numbers. But the overall point that's being missed here is that the way that you're building vivariums is that you're building them with the here and now in mind. When dart frog keepers build vivariums, we are planning on those vivariums lasting for years. Typically it takes about 4 to 5 years before we say, okay, we need to change out the substrate. We need to renew the substrate. Whether it be just taking out the old substrate, putting in new substrate, or most of us put in new substrate, and then yearly we typically put more leaf litter on top because it decomposes over time. Its nutrients are utilized by the plants, and then we cut the plants, so we take portions of the plants out. So that's our output from the tank, is the plants. Our input is every 4 or 5 years, more substrate, and then about every year, more leaf litter. But the way that we're building these vivariums, we're not just building them to last years, but we are also building them to be a stable habitat for our animals. We don't want a swing in humidity to kill our animals. We don't want a bloom in bacteria or a bloom in fungi to kill our animals. We want to build in buffers. And so, yes, that adds in more complexity on the front end to how we build those vivariums, but it is very cheap considering that that added complexity on the front end is going to make that vivarium last for vastly longer. And it's going to add in all of these buffers to ensure that if something goes wrong, which things will eventually go wrong in some capacity or another, then those things are in place so that your animals are going to survive. So it's more likely that your animal's going to survive. It's like I had a tank one time that, I had a frog that unfortunately didn't make it. And there were a couple froglets in there that I had no idea about because they had raised them up in there. I searched all through the tank. I could have sworn there was not a single frog in that whole tank. It was a couple of rediquitos. And so I went ahead and I closed the tank. I taped it up. I quarantined it for 6 months because I didn't know how that adult died. It was so badly decomposed I could not run necropsy on, unfortunately. And 6 months later, I came in and I looked, and sure enough, there was 2 frogs in there that were perfectly healthy, that had been feeding upon the microfauna in there. I've had tanks before, unfortunately, in the past that were overtaken by Berbamii fungus, the yellow flower pot fungus. And that was actually one of the main reasons why I wanted to develop this beneficial microbial inoculant, because I noticed that when I inoculate the soil with other types of fungi and bacteria, that was, yes, the fungus, the Berbamii would get into those tanks, but it was then slowed down exponentially. It was to where it eventually just fizzled out and went away, so to speak. The fungus was killed off. And the reason for that is because fungi actually wage mycological warfare. They have mycotoxins where they actually kill other fungi. So when you're putting in beneficial bacteria and beneficial fungi, you're actually colonizing that vivarium soil with beneficial things that are then going to kill off anything that's harmful that's going to overtake your vivarium. So again, I think that the way that we build vivariums should be more complex. It should be the way that nature is, which nature is very complex in of itself. And when we try to simplify it too much, we make it into something that is not going to be long lasting. I'm talking about years. And it's something that if something goes wrong, which it eventually will, then the animals are not going to fare well. Anyways, again, I really appreciate you having this debate with me, and I am having a little bit of fun with it. I hope everybody watching is learning a lot of things about this as well. And anyways, I think that unless you have anything else that you'd like to address with this, then we may even move on to the next topic and talk about drainage layers. So, thank you.